All right, it looks like we are live. Well, everyone, I'd like to introduce you to Pubtalks, and specifically, I'd like to introduce you to Ostello Bello, where we are hosting our evening, however virtually it may be. Imagine you hear the sounds of beer being poured, glasses being cheered, friends laughing, and many languages being spoken. We're gonna have several presenters, scientists, talk about their work tonight and share some of their passion for science with you. And without further ado, I think that we all need to open a little beer here. So if you have your own at home, please follow along with all the processes, including the most important one of having a beer as we listen. Without further ado, I'd like to introduce Giovanni Giangreco, who is a postdoc at the Crick Institute in the UK. And take it away, Giovanni.
Thank you, Corey. I have my beer here. I'm going to share my screen now. Okay. Can you see, Corey?
Yes, we can see.
Okay, cool. Okay, so first of all, good evening, everyone. And I would like to thank a lot the organizers for this event. I want to say that I was part of the organizing committee back in 2017 in Barcelona, and it's been a lot of fun and very tough, but also really a lot of fun. So congratulations to the organizers of this year event. So tonight I'm going to talk, as you can see from my title, of Cell Olympic Games. And in order to understand why I picked this title, I need to go a few weeks back. So basically, when Corey proposed me to prepare a science communication presentation, I was happy of doing this and I accepted immediately. And then I said this to my family and friends. I said, yeah, there's this opportunity. I'm going to present in a funny way my project, some of the things that I'm doing here in London. You may be interested to see, so you can have an idea. It's going to be interesting. However, differently from what I was thinking, the answers that I got were more like that they were expecting something boring like this one. So a very boring lesson with a lot of people sleeping or doing other stuff. And interesting, some of my friends especially, they said, not sure I can come because tonight there is Serie A in Italy, so I think it's more interesting. And this is fair, obviously. But then in the days after, I started to think, oh, so is really science boring? So why do people think this? So I think that especially in this time, it's clear that science is important for society and not only, but there's still different kind of stereotypes about boring lessons, long experiments, and this kind of, boring environment. While, at least from my perspective, science, especially when I was a teenager, I do recognize science and scientists more with the kind of other two figures, like Doc in Back to the Future or Data from the Goonies. So because of this, and because of basically the answer that some of my friends said that they prefer to see Serie A, I decided to organize Cell Olympic Games because I am expert in cells. I work every day with cells, and because I thought it was worth to organize an athletic event in opposition to people that will see Serie A tonight. So welcome to the first Cell Olympic Games, and this year is the year of the Olympics, so it makes also a lot of sense to organize an Olympic in this year. And I hope that the aim of today for me is going to be to show you that science and scientists try to ask important questions also in a funny way, but these kind of funny way questions that are also going to be used to answer important questions. So the first competition that I want to present you today is the 100 meters race, or probably more properly, I should say, the 1,000 meter race. So in this kind of competitions, scientists from all over the world chose, I think, 54 different cells. So each of them that you can see here is a cell. So they basically added these cells on top of linear tubular structures, and then they imaged how they move and how they spread over time. And as you can see here, these kind of cells have different properties. So some of them are very fast, like this one. Some of them are very slow, like this one. Some others tend to be persistent in one direction. Some others instead tend to. So this was the first screen that they used, obviously, to characterize the athletic parameters of cells, which are basically these physical properties. But my take-home message for tonight is that scientists are not only doing boring things, but also very fun things. So indeed, after this first screening, scientists decided to organize a real actual world race in which they picked 10 different cells. They also had a starting line and a finishing line. And I think it's worth to say that of these 10 cells, then the winner was this one, which is a human embryonic cell. Now, this is very fun. Listen my opinion. So is this interesting or not? Also for non-fun reason. Well, I guess you can imagine, but indeed, this kind of information is important. For example, during metastasis acquisition, indeed, cancer cells in a primary tumor need to spread and to move from the primary site. Then, for example, they can go into the circulation and they can extravasate and give rise to metastasis in secondary tumors. And scientists understanding the parameters of these cells, which means their persistence, their velocity, the mean velocity, the maximum velocity, is definitely informative for them in order to understand the mechanisms behind metastasis acquisition. Now, the second competition that I want to present you today is the 100 hurdles race, which is, as I hope you understood now, probably 1,000 micron hurdles race. So in this kind of competition, researchers have designed a microfabricated device, as you can see here. Now, very briefly, on the left side, there are cells that here you can see them in red, while on the right side, in this kind of box at the end of the device, researchers add some kind of molecules, which are called chemoattractants. So now, this kind of chemoattractant molecules have the tendency to attract cells. Depending by the type of cells, by the type of molecule, by the concentration, cells will be attracted more or less. And obviously, also in this kind of setting, in this kind of system, it's possible to create a competition, as you can see here, and to calculate which type of cells is attracted more or less. In order to mimic a more physiological environment, the researchers decided to have ice with these dead ends, which mimic somehow the hurdles of a hurdles race. And so the take-home message of this is, why is this important? Why scientists are trying to do this kind of setting, this kind of device, and to measure the efficiency of different cells in order to reach, for example, the box at the end of the device? Well, so chemotaxis, which is the process that describes the cells that can be attracted by chemoattractants, is important in many biological processes. For example, and this is a movie from the Weizmann Institute, this is important, for example, I was saying, during embryo development. So this is a chicken embryo, and in green you can see that some cells that at the beginning are in the center of the embryo, but during the development they move towards the end. So they know how to go through the edge of the embryo. And in this kind of physiological process, chemotaxis is very important. It's known that is very important. And these kind of devices are trying to mimic somehow the properties of some of these cells, and then to try to understand how the cells behave and sense the environment. The third type of example that I want to present you today is the hide-and-seek game. I'm assuming that all of you know the hide-and-seek game. The actors that I'm going to talk with you tonight are two. So we have cells that are in green, which is the hiding cell that doesn't want to be discovered, let's say, and we have the seeker cell that is in cyan. Now, the other piece of information that I need to give you, and this is a particularly interesting competition game because it's something that I'm studying now at the moment here in London. So in order to understand this game, I need to tell you that if we take cells, and this, for example, is a cancer cell. So if we take cell and we let them stay in a plastic, like scientists do every day in every lab in the world, so the cells will start to spread and to proliferate. So over a few hours of days, they will have filled basically the whole space. So that's the starting point. However, if we add a seeker cell in this kind of game, which is probably a very cruel game, so when the seeker cell recognizing the hiding cell is going to die. So let me give you an example here. So as I hope you can see, so here we have the hiding cell in green, the seeker cell in cyan, and these are both of them together, as you can see from the bright field image. So compared to what I was showing before, in which there were only hiding cells, here with the addition of this kind of seeker cells, these seeker cells are very efficient. They spot immediately the green cells, the hiding cells, and as I hope you can see, they start to create this cluster here and they die. So basically, the green cells are not able to fill the space, so they do not proliferate anymore, and they are dying. Now, this is probably interesting to see. However, I think it's much more interesting when I say that in this kind of setting, the green cell is a cancer cell and the cyan cell is a lymphocyte. And this is a classic way of immune control over cancer cells. And this is something that we try to study a lot in order to understand how immune cells do recognize cancer cells. What is important, indeed, there are some other cells. These are other kind of cancer cells, always in green. In this example, I also added lymphocytes, which are the seeker cells, but these cancer cells are able to hide very efficiently, and indeed, they do proliferate a lot, and over the course of time, they fill all the spaces, and suggesting that in this kind of setting, this kind of hiding cell is very strong and is able to escape the seeker cell control. Now, that's the system that this kind of cruel hide-and-seek game is what I'm studying now at the moment here in London, and is important for many processes, especially in cancer, as I hope you have understood, and is at the basis of the immune therapies. And this kind of system helps scientists all around the world to understand how immune therapies works in cancer patients and how cancer cells can exploit different type of resistance mechanisms in order to evade the immune control. Okay, so for today, the take-home messages are almost two. The first one is that I hope I convinced you that science is much more than boring experiments and lessons. The second one is that competition games are something that scientists do use every day in laboratories to recapitulate and to try to understand important physiological and pathological processes. And the third one, for the people that I'm expecting eventually will think that hide and seek is not really an Olympic game. Yes, this is true, I know, but I think that I've convinced you that this evidence suggests that it should be added in the next Olympic Games. Now, the last slide is that I want to thank the people that helped me to are helping me here in this project, in particular my supervisor, Eric Sahai in London, all the members of the tumor cell biology group, the people that work at the Francis Crick, my funding agency, which is the Francis Crick Institute itself, the MSD, and I want to thank you all for the attention.
All right. Really nice talk, Giovanni. Thank you.
Thank you. I'm not sure if I stopped shared.
You did. Now we just see your lovely face. So I have a question, maybe more of a curiosity, but my helper wanted some screen time. Giovanni says hi. So the age-old question of who wins the race between the turtle and the hare. So is faster better, or is there also some slower methodology that cancer cells may use to get to a certain place, to turn the right corners, to follow the right chemoattractants?
You mean fast as a speed or fast as a proliferation?
Speed of movement.
No, yeah, I guess that also the cancer stem cell theory would suggest that it's not about proliferation and migration itself. It could be that there is much more. So I don't think that the faster is the one that wins. But it could be that the faster is the one that starts the run, and then others could exploit their way of moving in order to improve, for example.
Okay, so they follow one another, and they kind of they pave the way further.
Yeah, that is also something linked to for example, to the cluster of circulating tumor cells. So it seems that at the beginning they start as single cells migrating and going into the blood system, and then are probably the clusters. At least this is one hypothesis, that are the ones that are metastatic, that have the more metastatic potential. So there might be some link with the speed of some cells, or but that's just an hypothesis.
Cool. So Audrey Cobetta asks, how do you make the cells green or blue?
So we take advantage of a discovery done a few decades ago. It won also the Nobel Prize. So we use, so for the green, we use, it's called the green fluorescent protein. We basically use different fluorochromes. These are proteins that have the natural tendency, when they are excited with specific wavelengths, they have natural tendency to emit in other wavelengths. So we have, let's say, scientists over the course of the decades have discovered multiple type of colored proteins. They can be cyan green, they can be red. So there's really a lot of creativity about this. And so we basically insert a protein that is naturally able to give fluorescence. The only thing I can add is that this is coming from, the first one, if I'm not wrong, is coming from a jellyfish. So in nature is there are some animals that are able to be naturally be fluorescent. So basically scientists try to understand how, and that's where everything started.
Very cool. Thanks. Colin Miko Jones from Phoenix, Arizona, I believe, asks, which cells were hider and which cells were seeker cells? Was this like immune cells used?
Yeah, so that's true. So the seeker was an immune, so it was a T cell, and the hider were cancer cells. And like in this example, I'm taking advantage of a classic model system that is used in our field, which is antigen recognition with the albumin peptide, and this helps us to understand how T cells, for example, are able to recognize cancer cells or not.
Nice. Thank you very much. I think that is unfortunately all the time we have for questions for Giovanni. But if you guys, hold on, we have a last two questions here that are asked in the eVideo stream from Ted. Is there a race of humans where some go backwards and some go forwards? I think that was more rhetorical. But the real question was, if you put a fast cell and slower cell on the same track, what happens if the slower one is initially in front? So do they leapfrog? Does the fast one get stuck?
Yeah. So the short answer is that I don't know. I've not done this, so I don't know. I would say maybe it depends by the type of cells. So there would be probably some of them that are able to, I don't know, like go on top of them. Others probably would stop. Yeah, I don't know. I think it would be interesting.
Cool. Thank you very much, Giovanni. Everyone, let's give him a virtual round of applause. Clap, 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 clap.
Thank you, Corey.
Thank you. All right, moving on to our next speaker, Judith Hariprakash. She's actually my colleague here in IFOM in Milan. She's towards the end of her PhD, I believe, and she's a bioinformatician. So, without further ado, Judith, take it away. Oh, wait, sorry. Before things begin, I didn't mention it last time. Guys, if you have any questions and you're coming from YouTube, feel free to ask questions on the YouTube chats, or you can ask questions if you're coming from the meeting on the Enable chats. If you just type your questions there, I will give them to Judith at the end of her talk, and then we can have a nice little discussion. Thank you. Judith, go ahead.
Thanks, Corey, for the introduction. I'm so happy to be here. I'll be talking to you about decoding life in time and space. Okay, so some time ago, when things were normal, we had these two kinds of people. We usually classify them as introverts and extroverts. And this is basically based on the way the people interact in a social setting, like whether a person enjoys their solitude more or takes up energy from being alone. We classified them as introverts. And those people who enjoy socializing, who like to gather, to be around people, who take up energy from the environment, were classified as extroverts. So in today's talk, I'm going to introduce you to a group of introverts and extroverts that are present within ourselves. So who are these? So before going into introverts and extroverts, let's just have a look as to from where did we come from. We all know we came from a single cell, and this cell multiplied time and again over months and years, and we became who we are today. So, and this cell has a DNA that we inherited from our parents, and that's the reason why we are lookalikes of our parents sometimes, or sometimes we do the exact same thing as a parent, or maybe a sibling, or even sometimes in some cases we do certain things exactly like our grandparents because we have something very common between our parents. The DNA is inherited. But wait a second, isn't it that we came from a single cell? And if that's the case, all the organs that are present in our body is from that same single cell. And if this DNA is from our parents that we got, it's present from the single cell, the same DNA is present in all these organs. But how does this differentiate? I mean, how does a lung know that it has to do a specific function? How does a skin know it has to do a specific function? How does this all happening? So if I actually look into this, the DNA basically has regions called genes, which gets expressed into protein. So this mechanism is what kind of controls the functioning of each of the cell and thereby the organs and thereby the entire organism. But this expression of a gene to protein is actually tightly regulated. When I say tightly regulated, it is more like an orchestra when things perform very synchronizedly and everything is controlled and only certain things get expressed and certain things do not get expressed. And this organization is actually multi-layered. The DNA is basically just the information that is present. Well, on top of it, there are multiple layers of regulation, and this is what we call it in scientific terms as epigenetics. So epi is from a Greek word meaning on top of it. So because these layers are present on top of it, we call it as epigenetics. So one of the things about this epigenetics is packaging. So basically, DNA is around 6 meters long, while it is packed into a nucleus, which is only 3 micrometer in size. So it's almost like packing so much of stuff into a small suitcase. But you know what? If I am doing the packaging, if you give me a 6 meter thread and ask me to put it into a 3 micrometer nucleus, I'm just going to stuff everything in. And when I need to use the thread, a part of the thread, it's going to be very difficult for me. I may have to take it entirely out and then do it. But then that's not how the DNA works. That's not how a cell works. It's not like when a cell has to work, the cell splits open, all the DNA comes out, and then it does work. No, the cell does it, everything within the nucleus. So how does this does is very interesting. So this is where I think all the architecture people have to take a note on as to how the DNA is packed into the nucleus. So the DNA, which is a thread-like structure, is wrapped around protein called histones, more like a bead, and then you wrap string around it. So this is actually called a bead on string structure. So these bead on strings are further then coiled, more coiled and more coiled to make into certain compartments. And this wrapping around is not just done, okay, I will just wrap everything around and stuff it in. No, it's done in a way that is not just looking, when you look at it, it's beautiful, but it's not just beautiful, it's very functional. So in this one of the functioning aspect, if you see that certain part of the DNA is loosely wrapped and certain part of the DNA is very tightly wrapped. And these are called open and closed chromatin. So this is where the introverts and extroverts come into. So the open chromatin, which is basically loosely wrapped, are open, and therefore they're free for socializing. Basically, they're free for expression. So when other proteins come and interact with them, the region where it is open has whatever genes are present in them gets turned on, which basically means the gene is expressed. Whereas the region which is like closely packed are similar to your introverts, as in they're confined to themselves and they do not have other space for others to come in. This is the regions which are turned off. So this is one of the mechanism as to how your DNA is placed in your nucleus in a cell determines which part of the DNA to be expressed, as in which gene to express. This is just one layer. A person being extrovert doesn't alone mean that the person will go out and party every day. It depends on if the person is having the mood to do it, if the person is available, or the place where they want to go and party is accessible. So it's similar to these other things that determine if a person is going to a party or not. Exactly, if a gene is going to be expressed or not also depends on other layers of regulation. One is a chemical modification on the DNA. Something called a methyl group is added to the DNA, which determines if the DNA is going to be expressed or not. Same, the proteins that I talked about before called histones, that the bead onto which the string is wrapped, even there's some chemical modification on them is affecting if this region is going to be expressed or not. Similarly, accessibility, if the region is open, there has to be proteins that come and interact that are freely available in order for this expression to happen. So these are the multiple layers that are present in this regulation. And this is what I'm trying to study in my PhD. But why should you care? It's nice to know, but why do you care about it? Because what scientists have recently found out is that the diseases that we see today, such as cancer, diabetes, cardiovascular damages, are not just because of the mistakes in the DNA that is leading to these diseases, but also some problems in the epigenetics is also leading to such diseases. So if that's the case, what should I do? So people have also identified that some of the environment, the lifestyle changes that we do may impact the way your epigenetics is working. So it's not like a straight line that if you do this, you're going to be healthy. But then if you take care of your body is going to take care of you. So with that, I'm ending my talk, and I would be happy to receive some questions.
Very nice talk, Judith. Also, as a side personal note, I always love bioinformaticians talks because you guys have the best visuals. You have the best slides, the best transitions. Very nice, very beautiful. Apart from the beauty of it, also the content was super interesting. Thank you. I have a personal question. You show nuclei is fairly compact, very full structures. But I guess at a certain point, the DNA has to turn to RNA, proteins have to move around there, DNA has to replicate as well. So how much free space is there? How much of that is actually completely packed with material, and how much is just air or other stuff?
Okay, I do not know the exact numbers as to how much space is left, but then there is a certain amount of space because of wherein the DNA is converted into RNA. But then when it gets converted to protein, it goes out of the nucleus, and there is a space called the cytoplasm where you actually, where most of the because protein is a bigger molecule and it doesn't fit inside. When it has to get synthesized, it happens in the cytoplasm. So the part of the DNA doesn't come out per se, but then the product of the DNA travels around. There are spaces between the nucleus and the cytoplasm. There are holes there for it to travel from here to here, and that's where you have these things. So I'm not sure of the exact space. Maybe I should find them.
No, yeah, sorry, that was a very specific question. Is there a way to convert introverted DNA to extroverted DNA, or vice versa from our doing? Like, is there some medicine that we know to do this?
Not exactly, because but right now this phase of our research is to try to understand which part of the DNA is introverted and which part of the DNA is extroverted. This is the kind of research we're trying to identify. But then certain amount we're able to change in terms of probably doing the modifications and trying to see if they are becoming more closer or not. But then as a drug, I think there isn't any, but then it would be nicer in the future to have such drugs to change the entire structure. But then there is no clear drug so far, at least to my knowledge.
Okay, thank you. Ted is wondering, what is your favorite histone modification and why?
Okay, my favorite histone modification would be 27 acetylation. That's because it is a histone modification that actually determines if the region is going to be open or close, and that's the reason. I mean, it determines a lot of things. If a gene is going to be expressed or not is determined by this histone K27 acetylation, and that's the reason. And that's a histone mark that I'm working with, and maybe that's the reason why I like it too. Maybe
a little bit biased.
Yeah.
Sorry, that question was actually by Linda Krause. I don't know why I said Ted Weiner. Forget that. Waiting for any other questions here.
She said.
All right, I think that's all we got. If you would like to leave any last parting words with our audience, or maybe you concluded already.
Yeah, stay healthy. That's all we got to say.
Fantastic.
Thank you.
Thank you so much, Judith. Let's give her a virtual round of applause again. All right, we are bearing down here on our very last presenter of the night. I am also getting towards the end of this beer, as you guys can see. I hope you're all in the same position. So the last presenter is Ted Weinert, coming directly from Tucson, Arizona. Should be nice and hot over there, but hopefully he is not. All right, Ted.
All right.
So again, yeah, we can see you, we can hear you okay.
Okay,
so cool. Again, guys, if you have any questions during his talk, please leave them either in the YouTube chat or if you were coming from the meeting, leave them on the meeting chat next to the live stream. And take it away, Ted.
All right, so you can see me and you can hear me? Oh, wow. So thank you very much. Giovanni and Judith, you guys have set a very high bar for me to try to wow these beer drinkers at a local beer establishment, but I'll do my best. Start off by saying I'm not very artistic, and so I apologize in advance for some of the hideousness of some of my slides, but hopefully you'll get the point, because the message, I think, is very cool, and maybe it's the most important message in biology. How's that for a challenge? Okay, and thanks to the organizers. Anyway, here we go. Okay, so the title, there's two titles. Chromosomes, the Houdini of change. You'll understand what that means in a second. Houdini was a guy who was an illusionist in the early 1900s that used to do things that would wow people. And you sometimes you didn't see the change, but it was he was an illusionist. And I'll tell you what that has to do with chromosomes. And then the other title is imperfection. And you'll know all about those by the time I get done in 12 minutes. Okay, so chromosomes. Let me just reiterate maybe a few things that Judith said and set you up for this story. So chromosomes are illusions of no change. They actually do change, however slightly. So this is, look on the left, you can see my laser. There's the first cell of you. Your mom and dad got together, they made you. In that first cell, you had a chromosome. Let's say it looks like this. You then, that first cell grew, and I'll tell you more about this in a second. It divided and divided, made more cells. That was a young you with millions of cells. More and more duplications occurred, and there were yet more cells, and there was an older you. It turns out that the chromosomes in the older you look very much like the chromosomes in the younger you, except for a few things we're going to call blunders. These little red lines there, those are mistakes, errors, change that I will talk quite a bit more about in the next 11 minutes. These rare changes are not a problem generally for you, okay? But they are extremely, the process of forming these blunders is extremely important for evolution. Blunders are crucial to evolution. Okay, and this is going to seem a little counterintuitive, that imperfection is actually so important in biology, but that's the message. My favorite quote is from a biologist author named Lewis Thomas, who said, "The capacity to blunder slightly is the real marvel of DNA. Without this special attribute, we would still be anaerobic bacteria and there would be no music." I would like to thank him for that quote. I love it. I always use it in my biology classes when I teach. I might edit it by saying, without this, if we did not blunder at all, there would not even be anaerobic bacteria and there would be no music. And you'll understand why I'm editing that on the last slide, perhaps. Okay, blunder slightly is critical. My favorite idea is summarized in the next two slides. And it's really not a theory anymore. It's really a fact. And here's the graph. I'm a scientist just like Judith and Giovanni and Corey and lots of other folks. We love graphs, okay? They can summarize lots of information in a short little bit. And this graph, it summarizes my entire next 10.5 minutes. On the x-axis, you have chromosome change, blunders, etc., from rare to some to often. And the y-axis, you have forms of life from very few to lots. Okay? If I had time then, which I don't, I would ask you with your beer drinking partner to guess what the curve is going to look like. Would it look like this, like that? You know, what would the deal be? That plots chromosome change against forms of life. And the answer is that. It basically is a hill, as shown, where if there's very little change, if there's rare change, there's few or no forms of life. If there's some change, there's lots of life. This is where we are, thankfully. And if there's too much change, you either have too much life, or you have mutants, or you have cancer. Okay? So remember this graph, impress your friends and family, and hopefully you'll explain to them why this is such a big deal. Okay, so what is a chromosome? Well, what's a cell? I'll just give you a little background so we can dive deeper into this really cool idea. Here you are, trillions of cells. I like to say my 5-year-old kid drew this, but I did. That's as good as I can be. So there's cells in this person, trillions of cells. They have, I'll sort of expand this. Here's a cell. There's a thing inside called a nucleus, which Judith just talked about. There's chromosomes in that nucleus, and the chromosomes have DNA. The DNA makes tools that allow the cell to do stuff, as Judith also alluded to. All right, so cells, chromosomes, DNA, and tools. Those will be reiterated in just a second. So how do we grow from one cell to a body? Okay, this is important to understand the deal about blunders. So over here on the left-hand side, we've got our first cell again. When it duplicates, okay, one cell becomes 2. Each of those cells duplicate, so 2 cells become 4, and on 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 and on. Okay, all during these duplications and doing all subsequent duplications, slight blunders happen. All right, so you end up with 100 trillion cells, as it turns out, and you end up with chromosomes that have a few blunders. I call this the short tree of duplication, which will be in contrast to something I'm going to show you in a few minutes. This is not unique to humans. All biological organisms, here I'm showing our favorite next biological organism, bananas. Bananas do the same thing. There's a cell with a chromosome. It divides, there's slight blunders as they divide, and then you get a banana out at the end. There's a little artistic license here. Bananas and humans don't actually have the identical chromosomes, although they're not that different, as you'll hear about in a little bit. Okay, so slight blunders occur during this duplication. How do blunders arise during duplication? Right, and most of them occur by duplication. There's other ways to get blunders, but by duplication is the main way. So I'm going to use now this diagram. This is a chromosome, And these 3 boxes here are tools that do things. Those are genes that usually encode or frequently encode proteins. I'm just showing 3 on this chromosome. We've got 20,000 of those in our cells, so it's a lot. So the way duplication works is there's a duplication machine, a tool, which is this little very poorly written and these little balls of red, green, and black here, which are taking this chromosome here in this one cell and duplicating it to get 2 chromosomes. Okay, and usually this machine's really good at it, and it basically makes 2 cells that are virtually identical to the first cell. That's good. However, this machine somehow, this tool makes mistakes. It makes blunders, and those blunders are usually bad for the chromosome. They break it. Here's one that's broken. There's another one that's broken in 2 places, or you can have blunders where you just miss a gene altogether. This tool is gone. So that's how blunders arise. Okay, blunders can also be single letter changes. So here's our chromosome again. Here's the little black tool, and now here's a sequence. If you know what that means, there are 4 letters to the genetic code. And this, now I'm going to say this spells a word, let's say info, which will be relevant in the next slide. And if you have one base change because of the tool made a mistake, you get a mistake here and no longer spells the same word, inho, which is that's a broken tool. Okay, that's the analogy here. All right. But sometimes, luckily, blunders make new tools. So this is the mind-blowing aspect of evolution that I'm going to now illustrate on this slide. So here we have our beloved chromosome again, the 3 black genes that encode info, that encode rum, yum yum, and that encode nation. So all of those things make some sense, right? Rum maybe more than the other ones. And then what happens sometimes during blunders is that you don't make a broken chromosome, but you make a chromosome that has an additional new tool in it. So in this case, the black one, the blue one, and the red one are still there, but now we have a fusion, an additional fusion of black, blue, and red all fused together, and that now spells information. Information is not present on that first chromosome, but it is on this chromosome. This is a new tool. Okay, woohoo! This makes a better cell than that cell. All right, this process of blunders and making new tools is the way evolution arises. A cool example of a new tool is here's a chromosome and here's an ancient gene that encodes hemoglobin, which is this protein, the little black thing that binds oxygen. During evolution, there was blunder, and we got a new chromosome that had 2 of these black genes. Each one had a few changes in it, a few single letter changes, such that this one makes an adult hemoglobin that is then going through your body, okay, binding to oxygen. And this one makes a fetal hemoglobin. This is a woman with a little baby there in her tummy. And this hemoglobin binds oxygen in a different way than that one does. And that is really important for us that I can tell you about some other time. All right, so that's an example of a cool new tool. Okay, blunders in evolution and in you are similar. Okay, this is showing the slide I showed you a little while ago. Duplication, usually it's okay, sometimes you get blunders. All right, so what's the difference between blunders in us and blunders in evolution? Well, it's a simple, quote unquote, issue of the size of the tree. For us, there's very few cell divisions, there's very few duplications, and the blunders don't make much difference to us. In evolution, this duplication tree is really long. Maybe it's more like a forest than a tree. And what happens is when one cell becomes 2, becomes 4, and the number of cell divisions, the number of duplication cycles is not infinite, but nearly so. There's blunders happening all the time and new tools are being made. Okay, so it's the length of the tree that's the difference between us and evolution. but the basic process of blunders is the same, right? And you go from a simple chromosome with a few tools on the left, blunder, blunder, duplication, blunder, blunder, to a much more complex chromosome with lots of tools, right? And that, ladies and gentlemen, is evolution in a nutshell. Note, 50% of the tools in humans and bananas are the same. Okay, so this is out of the web. At my house, we have a little magnet on our kitchen refrigerator that says that humans and bananas share 50% of their DNA. Get over yourself. But that's an aside. Lots of cool questions remain. Why are duplication tools imperfect? We don't really know, but we are happy for the imperfections. Notice the way I wrote this error-free. My favorite is that there's no selection for perfection, but that's a matter of debate about why the duplication tools are not perfect. Do such blunders really occur? You know, we're inferring much of this from current, looking at current cells. Well, it turns out these blunders are very easy to duplicate in the laboratory. We do in my lab, where you take a yeast cell that has a blue, a red, and a green bit that doesn't actually say anything very interesting. We allow it to duplicate and blunder, and then we get something out which is Ura3, which means something to us yeast geneticists. Okay, so we did this in a study that was published a few years ago. So lastly, celebrate imperfection. Don't be fooled, chromosomes do change. We exist because of blunders. So go forth and blunder. And remember this figure, and you will wow your friends and neighbors. And that's it. Watch out for mutant bananas. Thank you.
Well done, Ted.
All right.
Very nice job. That was really fun. Actually, I blundered the introduction at the beginning of this. I said you were from Arizona, but I did not mention that you are a professor and you've been running your own lab for 30 some years.
Yeah, that's true.
Yeah, so
that's all true.
You know, nothing to scoff at there. So
there
he's not talking out of the rear end, as we say.
Yeah, I see.
All right. So the first question we have is from Linda Kraus. And she was asking, how does the new tool get to my daughters and sons? So I guess she's referring to her own tools and her DNA. How do they get to her daughters and her sons?
Let's see. How does the new tool? Well, it goes through the germ cells, so through the sperm and the egg. So the only new tools that I'm not sure if this answers her question, but here's the answer that I can come up with. New tools that form in us are only formed in our germ cells. Can you see this?
Yep.
Okay. Germ cells are right in here, right where the gonads are and stuff. The new tools that form up here don't go to your offspring. Only the ones that form in your germ cells. You know, if they're there in the DNA, they end up in the they end up in the first cell for your kids, and there they go. New tools are extremely rare, you know, for us to look in current day situations. So like Corey, for example, you don't really have any new tools probably from me, even though you're my son. But if you took me to you, expanded bazillion times, then we see some new tools. All right.
Okay. Nice.
I don't know if that answers the question, but that was my try.
I think it did.
Okay, cool.
Linda, if you're not satisfied, feel free to answer a follow-up and maybe you can stump him. So Valentina Feiner asks, over time, from YouTube, time, from anaerobic bacteria, we became humans. So what do you think we're going to become in the future?
Oh, that's a good question. I hope something even better. How about something apolitical? We would all be one. We would realize it. We would realize we all come from the same set of blunders, and we would stop arguing with each other. That would be a good outcome.
So we will be one giant amorphous blob of thought and consciousness.
There we go. There we go. And happy communicating people. Yeah.
Happy communicating people. Nice. Bonnie Colby asks, do biologists try to predict blunders or to create a model of the patterns of blunders?
Yes, I don't think. Yeah. So you can predict blunders by looking at the DNA sequence. You can predict what blunders are going to happen, and then that's actually what we did in that slide I showed you. You can actually then make a chromosome that will do the blunder. Okay, so you set it up so that if it blunders, you can know it because it's saying you're 3, which is a small molecule. So yes, you can set up chromosomes to make blunders, and everybody, there's lots of people in my field, yeast geneticist type people that do this kind of thing. So yes.
Cool. Engineered blunders.
Yes.
Giovanni Giangreco is asking, what about the whole 3.2 billions of base pairs of genome that we have? Is it partially junk? If so, why has evolution selected for all of this non-translated junk?
Excellent question, Giovanni. Ha fatto bene. So this is a great question, and there's 2 explanations for the 3.2 billion base pairs of our genome. And let me just tell, for people who don't know, about 95% of it does not seem to make a gene, does not seem to make a tool. Okay, that's the current view. And so what's the 95% of this doing? And there's 2 explanations for this. One that I ascribe to is that it's junk. Okay, it's not doing anything. Most of it is actually transposons that are degraded. They're like burned out cars. Okay, and they're sitting there not doing anything, and we can't really get rid of them, so they just sit there. That's the junk thing. The other one is that they encode stuff. There are tools that we don't know about yet. Okay, that's in this 3.2 billion base pairs of genome, and there are examples of genes that we now recognize as being tools in that junk. Okay. But my view is it's still mostly junk. Okay. So yeah.
Nice. So if we got rid of that junk, would we be better off?
Would we be better off? So that's a great question. And I guess the answer is I don't know. I would think that, so it turns out that the duplication of 1 cell to 2, it's actually not limiting. If you actually make the DNA have less DNA, you would probably still duplicate with the same rate. So it should not, removing the DNA really won't probably help that much, is my guess. But that's just my guess.
Interesting.
All right.
All right. We have a few more questions still, actually. I mean,
okay.
Why not keep going? We're at the end
All
here.
right.
So Linda's also asking, are the 50% of our DNA that we share with bananas part of the junk or the genes, or both? Okay, so as I tell all my first-year students when they ask me a question, there's 2 answers to that question. One is I don't know. 2 is my guess is that it's the genes. Okay, 50% of the genes are shared between banana and humans. And the junk tends not to be as shared. I mean, it's still junk, but when you look at the actual letters, you don't recognize them as being that related to each other. Okay, so it's the actual genes of bananas that are shared with humans, which is kind of mind-blowing. Valentina had a comment here. I think maybe you can see it on your screen. Yeah. She said, "Thanks for the great talk. I wish I had learned genetics this way." Well, the good news, Valentina, is that he is still a professor at the University of Arizona, and he is still teaching. So you can still go to his classes if you want. There we go. That's exactly right. That's exactly right. Giovanni then had a follow-up question, actually, on his 3.2 billion bases of junk. Okay, good statement. Come on, Giovanni, let's do it. So he's saying, isn't it already enough of a function, the fact that having so many bases helps decrease the mutation rate of coding sequences? So I guess the point is that junk a buffer for mutations in useful stuff? Excellent question. My answer would be, one is I'm not completely sure because we don't completely know. But what do you know? My guess is that the mutation rate in the genes is independent of the mutation rate in the junk. Okay, so
it would be kind of cool if all the mutations and all the blunders were to go to the junk, right? That would be kind of neat. But as far as we know, that's not the way it works. The blunders are equally distributed in junk and in genes. So, but then, you never know. I've never heard this thought before. So it could be that there's something we don't understand that would lead to more blunders going to the junk than to the genes. But from what we know about mutation, I wouldn't think that would be true. Okay,
nice.
That's fun.
I think we've got just one or two more questions here, and then we're going to wrap it up because I think we're going pretty far with these questions. So Bonnie Colby again is asking, so our junk is what makes us unique and differentiates us from bananas?
The junk probably does not make us unique because the junk we think is not doing anything. It's the genes. Our genes make us different than bananas. Okay, that's the most likely explanation.
Good. So we are not defined by junk. That's a
right.
reassuring thing to know.
That's right. We are not defined by junk. But there's actually the Y chromosome in you males out there at the bar. The Y chromosome is mostly junk. There's only 20 genes on the Y chromosome, whereas normal chromosomes have 1,000. So most of your Y chromosomes, gentlemen, is junk. Discuss. Anyway.
I won't touch that one.
There we go. Good idea. All right.
Thank you very much, Ted. Dr. Weinert.
All right. Thank you, Dr. Jones-Weinert. Whoa.
My pleasure to have you.
There we go. It was a pleasure to be had.
All right.
All right.
All right. I think that wraps up all of the questions and all of the presenters for the night. So I'd like to remind you all that for those tuning in from the meeting, that the meeting starts again tomorrow morning, bright and early. Please come, enjoy the sessions. And I'd like to say thank you all very much for your attention. It was a pleasure to be your host. I wish we could have done it in person, maybe next year. Have a good evening. Bye.